Hello and good morning. As everyone said, I'm Xiao, and I will be talking about the effects, or what I studied this summer, the effects of chromatin and sex-specific aging with Drosophila lifespan assays. So aging is a complex process, and while research concerning the molecular aspects of aging exists, there remain unanswered questions concerning aging differences between males and females. Males and females share the same genetic information represented in different forms. This leads to very different phenotypes, and it is therefore important to study the possible causes for these differences so that we can better understand sex-dependent aging differences. The most notable aging difference is the observation that females live longer than males. This observation is true for many species with XY sex determination. For humans, females outlive males by 5 to 6 years. This is a substantial difference in lifespan. In this graph displaying United States male and female survivorship with age on the horizontal axis and number of survivors on the vertical axis, females outsurvive males at every age. Many theories on aging exist. They can be separated into genetic or environmental causes, such as gene regulation errors, diet, and behavior, like risky choices. This summer, I examined a possible genetic component to sex-specific aging. The main genetic difference between males and females is that males have one X and one Y chromosome, and females have two X chromosomes. In this image, the Y chromosome is to the right is much smaller than the X chromosome. The Y chromosome is gene poor, degenerated, and full of repetitive and non-coding DNA sequences. Organisms actively repress the Y chromosome and its features. Suppression mechanisms fail with aging. More specifically, heterochromatin, the compact gene poor region that is influential in transcription and transcription and chromosome segregation, is more present in male genomes. This unequal distribution of Y-linked heterochromatin has been shown to modulate phenotype and cause sex-dependent variations in gene expression. Heterochromatin levels have been shown to modulate lifespan and tissue integrity. The breakdown and loss of heterochromatin has been shown to shorten lifespan, and reversing this loss in aged Drosophila fly studies has been shown to provide genome stability and increased lifespan. These observations suggest that heterochromatin integrity plays a role in longevity and tissue integrity. And this link between aging and heterochromatin raises the question of whether sex-specific chromatin content causes differences in male and female aging. To test the effect of heterochromatin on the Y on longevity, I generated organisms with 0, 1, and 2 Y chromosomes. Drosophila is a powerful system because we can easily genetically manipulate them, unlike humans. Because sex determination in Drosophila is dependent on the number of X chromosomes, it is therefore possible to disentangle the Y chromosome from sex. In flies, the Y chromosome is responsible for sperm production, and males with only an X chromosome are sterile but phenotypically normal and viable males. If Y-linked heterochromatin plays a role in aging, then males without a Y should live longer than males with a Y, and males with two Ys should have the shortest lifespan. Also, females with a Y chromosome should have a lifespan similar to that of a wild-type XY male. To test these hypotheses, I performed lifespan assays. There are two crosses to examine the effect of maternally and paternally inherited X chromosomes in males. In cross A, wild-type females were mated with the mutant 2549 strain with attached XY chromosomes, and in cross B, strain 2549 females with attached XX chromosomes were mated with a wild-type male. Both these crosses result, as you can see, in females with the XXY sex chromosome composition and males with an X, only an X or X naught. In cross C, my third cross, the cross B XY females were mated with 2549 mutant males to create XYY males. The presence or absence of the Y and the number of X chromosomes was verified with qPCR, qualitative polymerase chain reaction, with primers targeting the Y, X, and autosomes to measure the ratios in the organisms. To set up the experiment, I collected virgin age-matched flies and separated them by sex. I placed them in vials with food with 30 flies of each sex in each vial, and the food was changed every 2 to 3 days. Environmental factors that affect age-specific mortality, such as temperature, food quality, and cohort density, were controlled for. The deaths were then recorded, and data was compiled into Kaplan-Meier survivorship curves, which estimates the survival function from lifespan data. The survival function captures the probability that an organism will live beyond a specified time to measure the impact of an intervention. It is a good method because it takes into consideration right-sensor data, in which participants leave the experiment, and in my case, flies flying away. I used wild-type males, meaning they had regular XY sex chromosome composition, as my control. The females outlived the males, shown in this graph. The percentage of surviving males drops precipitously faster than that of surviving females, despite males having higher survivorship during most of their lifespan. For the flies with an X chromosome maternally inherited, they lived longer than the females with a Y chromosome, with an extra Y chromosome. In this graph, female survivorship is below male survivorship for the entire experiment. In males with an X chromosome inherited from the father, it is also apparent that the males live longer than the females with a Y chromosome. This difference in male and female survivorship is present during the length of the experiment, and the difference is greater than that of the males with the maternally inherited X chromosome. For the males with 2 Y chromosomes, survivorship was lower than that of the wild-type flies. The graph shows that the XYY males plummeted around day 37, whereas for the wild-type males, they plummeted at day 48. When comparing males with 0, 1, and 2 Y chromosomes, the assays showed that males without a Y maintained 75% survivorship at 77 days and 74 days from cross A and B respectively, while males with a Y hit 75% survivorship at 46 days. The XYY males with 2 Y chromosomes hit 75% survivorship at 33 days. In comparing females with a Y chromosome to the wild-type males, the experiment showed that the XXY females had a lifespan similar to that of the XY wild-type males. The longer period of time for cross A versus cross B to reach 75% survivorship may be due to the fly strain I used. The mutant 2549 males with attached XY chromosomes have a different Y, a slightly different Y, which the cross A females inherit. The cross B XXY females, however, have the wild-type Y chromosome, and so I think that's my hypothesis for why crossbreed females has a similar lifespan to that of wild-type male versus the long lifespan for cross-A female. My next step is to perform replicates for these crosses and perform Western blots to quantify differences in heterochromatin between young and old flies and assemble total RNA libraries to measure differences in gene expression and junk DNA between young and old flies. I want to thank Emily Brown in the Backtrog Lab for being here and so hard to meet. I want to thank the Rose Hills Foundation for giving me this opportunity to do research this summer. And I want to thank Eric and Leah from SURF for their advising support. Thank you.